To the organizers for inviting me, and thank you so much for staying. It's been a great day so far. So my talk today is APOL1 and kidney disease in Africa, and I hope to give you some preliminary data from the H3 Africa collaboration. I'd also like to highlight that I'm really presenting on behalf of H3 Africa. So if you see other H3 Africa investigators, it's really a team effort, and I just happen to be the person up here. What's important to remember is that chronic kidney disease actually impacts globally, and that's really why we're all here. And you've heard most of the time during the talks, how do we actually improve survival here in Africa? How do we actually improve the care for kidney disease patients? And I think this was the campaign that happened in 2015, the Lancet Kidney Campaign, that really tried to show to the rest of the world how kidney disease is so important and how we really need to know how we can do better. What we've known in the United States, actually, for a long time, I was there before I moved to Canada, was that the cumulative incidence of chronic kidney disease and mortality in the African-American study of kidney disease was relentless. It was relentless whether you were treated with a medication of amlodipine or an ACE inhibitor, and it was relentless of whether you looked at overall mortality. That the majority of them actually progressed during the trial phase, during the trial and cohort phase, which is in the middle, and then the cohort phase at the end. So even with optimized therapy, optimized blood pressure therapy, there continued to be progression of chronic kidney disease. At that time, we actually started studying kidney disease and understanding the genetics that was related to kidney disease. But it's really important that we note that, in fact, all of us in this room, even though our skin color may be different, even though our hair may be different, our eye color may be different, that we're 99.9% exactly the same in our genome. And what we're trying to identify with a gene is what's that less than 1% difference that occurs between different populations to account for disease. When we embarked on the FIND study, the Family Investigation of Nephropathy and Diabetes, at Hopkins, where Linda Cow and I were, we were focusing in on non-diabetic kidney disease. And our hypothesis was that West Africans that came through the Americas through slave trade and the Europeans had significant admixture. And we would take the African-American population and take a look at the African genome among African Americans and determine who had kidney disease. And so it's important to recognize that this was sort of the hypothesis that we started with, that we would take 2 African chromosomes, 2 European chromosomes, and over time, with multiple admixture, you would eventually have the self-reported African Americans within the US that would have African genome and that would have a European genome. And our hypothesis was that we would find a gene on the African genome that could account for kidney disease that occurred among African Americans that was 4 times greater than what happened in the European Americans. So in this admixture mapping study, we identified the novel susceptibility locus on chromosome 22. It now seems in the dark ages, but we published it in 2008. And 10 years later, it's now sort of advanced a huge amount of work. We initially focused on MYH9, and the reason we focused on MYH9, even though APOL1 was right there, was that MYH9 had a Mendelian form of kidney disease called Fechner's or Epstein's anomaly that were associated with kidney disease. Data then came out, also from another group exactly at the same time, from Jeffrey Kopp's group showing an association with FSGS. And then a few years later was then the 1,000 genome data actually came up with some new data and an updated genome map. And Martin Pollock's lab was then able to look at MYH9 and APOL1, since they were right beside each other, and determine that it was in fact the coding regions of APOL1 that were associated with kidney disease. And biologically, it made some sense because it affects trypanosomal infection, so it could be analogous to sickle cell disease. The other thing was that it was associated with FSGS, similar to the initial paper. But the part that was so interesting was MYH9 is actually a gene that's conserved across species. APOL1 is not. So APOL1 is only shown in primates and is related to recent evolution. So there's clearly some significant difference there. It was also found by Carlos Kareki's group that, in fact, among the Ethiopians, there was also an association with APOL1 and not MYH9 with end-stage kidney disease. The hard part, and the only reason I show this slide, is that MYH9 and APOL1 are highly correlated. The only reason it's important is that I want people to recognize that this chromosomal locus is really important in selection that's occurred over the continent. It's unclear why. It's unclear exactly what the APOLs1 do other than HDL transport, but there's clearly something related to selection that has occurred in this entire chromosomal region. And it's quite important to understand the biology of this region as much as possible. So what's interesting about APOL1 is that there's actually the 2 haplotypes of APOL1, G1 and G2, which consists of 3 alleles altogether. APOL1 is bound to HDL and circulates. It's found in humans and some non-human primates. and it protects against African sleeping sickness. So if you have no alleles, such as that there was a person found with no APOL1, in fact, never got trypanosomiasis. But if you've got one risk allele, you have a heterozygote advantage that you don't get trypanosomiasis, but in fact, and you're not at risk for kidney disease, but if you carry the two risk alleles, in fact, you're at risk for kidney disease, but you still aren't getting an infection. So this was found is that this was probably the reason for selection, and all of us started focusing on APOL1. Many studies, including a number of studies done by people here in the room, really took a look at spectrum of diseases related to APOL1, and in cross-sectional analysis, either in case control studies or case-only studies, we're able to show that, in fact, this isn't really working, but if you take a look at the far right column, you can actually see that the odds ratios were quite high because they were doing case control analysis, typically with those who had end-stage renal disease. And if you took a look at case only, among the HIV patients, there clearly was a significantly higher odds of having kidney disease if you carried APOL1. In the summary study, it's really shown that in the green, you can see the case control studies are much higher odds, which is not surprising. And in epidemiological terms, we talk about winner's curse. And then slowly over time, if you look at the CKD cohorts or the population-based studies, the odds actually reduced to probably less than 2. And that's important to note because now you're trying to not just look at the odds, but actually relative risk and really see what the population attributable risk is. And as you take a look at larger cohorts, it isn't surprising that the risk has continued to come down. And the reason this is important is that if you take a look at this figure where I plotted some of the risk over time, depending on what age you're studying, what population you're looking at, you can actually see that the risk actually varies quite significantly based on the study population, based on the outcome that you're looking at, albuminuria versus chronic kidney disease versus hypertension versus end-stage renal disease. So it's important to note that you see significant variability in the studies, and the reason you see significant variability is because the outcomes differ, the population selections are different, and whether it's a case control or a population study. So the question is APOL1 really analogous to sickle cell disease? So we recognize that sickle cell disease gives you acute crises. APOL1, though, gives you progressive disease. They're both autosomal recessive. Sickle cell is actually complete penetrance. APOL1 is actually variable penetrance, which is really complicated because really you're not at risk if you carry one variant, but you're at risk if you only carry 2 variants. The other thing is that the infectious rate and the carrying rate of APOL1 versus sickle cell disease among African Americans is quite low among sickle cell, but it's very, very high among APOL1. And among European Americans, it's absolutely absent. So it's very important to recognize that even though we think that this may be analogous in terms of the story to sickle cell disease, what's very interesting is that it's actually much more common than sickle cell disease. The variant is much more common in the US, and it's 1 in 10 in the population. Yet not 1 in 10 have kidney disease. So it's important to recognize that it may be involved in selection related to infections, maybe some protective effect that it has, but still we need to understand what that is. So even though APOL1 explains 70% of the excess risk in end-stage renal disease, and even though the frequency is quite high, and even though we can show the association is probably 2 times more likely to progress with chronic kidney disease, we clearly need a second hit. We need a gene-gene or gene-environment interaction, or potentially both, to actually account for why those people who are carrying APOL1 are actually going on to develop kidney disease. So what's really important is actually when we first did the study in FIND and we presented it, somebody said, do you know that the West Africans are actually at risk for kidney disease? And at that point, there weren't many studies that were done, and I couldn't really answer that question. But I think now what we know is that really we have to study this within Africa. And what can be learned? Well, it's really important to recognize that the populations of Africa harbor the greatest human genetic diversity following an evolutionary history. That there's signatures of selection which are important, cultural practices, infectious epidemics, migration, admixture, or chance alone. And what's really also important is that now a paper that came out in 2014 showed that the burden of chronic kidney disease is estimated to be about 14% in the entire population. So if you think that there are 1 billion people on the African continent, 14% of them have kidney disease at this present time. That's an extremely large burden. Therefore, if we can't screen earlier, if we can't try and understand who's at risk for disease, how are we actually going to manage those many people? I think you said last night there was only 700 people here at the meeting, but there are potentially millions of people with chronic kidney disease, and there's not enough of you in Africa to handle the millions and millions of people who've got chronic kidney disease. And so we have to do this a little bit better to try and understand what leads to disease, and potentially can we actually decrease their risk. So when we try to understand who gets chronic kidney disease, we need to understand that there's genes, there's environment, and ultimately the disease we're looking at itself. And you can't look at one without the other. So I'm going to go over some work that we've done in the H3 Africa collaboration. Dr. Adu is actually the PI from Ghana. The work I'm going to present is actually part of the NIH-funded and Wellcome Trust collaboration. We have lots of sites within Nigeria and Ghana. Those are our primary sites. And these other sites are really related to other studies. What's important to realize is that over 70,000 people have been recruited through the whole H3 Africa collaboration, and these are all the countries that are currently involved. Again, as I said earlier, we couldn't do this study without all our collaborators, and it's really been a team effort, and I'd really like to thank the people from Ghana and Nigeria who've actually collected the 8,000 participants. So in our study population, what I'm going to talk about today is we currently have 3 grants that are funding the study. Can you see this? No. So we have 3 grants. I'm going to actually work on the first one, just talk to you about the APOL genotyping. But just so you know, coming down the pike, we're going to have a progression study on chronic kidney disease. We're also recruiting HIV chronic kidney disease, 600 children with nephrotic syndrome across the continent, and also 2,200 with sickle cell disease. But now I'm going to focus on the case control disease, which was recruiting patients with chronic kidney disease and comparing them to healthy controls. So why are we interested in the allele frequency of APOL1 in Africa? Well, if you just take a look at data that's from the 1000 Genome Project, so not as large as our 8,000, it really describes that the APOL1 frequency ranges from 10 to 50% in West Africa and less than 25% in East Africa. But what's very interesting is that trypanosomal infection is actually much worse in East Africa than it is in West Africa. And so even though the APOL1 is supposed to be there selected for trypanosomal infection, there's clearly something wrong with that pattern because it's not in the place where you think trypanosomal infection is actually endemic. The other thing is that it actually protects you only for one type of trypanosomal disease and not for all types, and I'll get into that later. This is also reported that this is the frequency of APOL1 risk variants, and it's actually so much higher than we see within the United States. Some studies have actually described them as high as 70% among West Africans carry the APOL1 risk variants. What's important to note, though, is that East Africa has a much lower rate and European Americans are much lower. What I think is fascinating, though, is when you take a look at this study that was just done by Don Stanifer, published in PLOS ONE, it actually took a look at a small subgroup that he had recruited within Tanzania in the first row, and he had taken a look at the Bantu, ancestry, Nilotic, Kosian, because basically your language actually determines your ancestry, and then took a look at the different tribes and was able to show that, in fact, the frequency of APOL1 genotype varies. APOL1 G1, G1, and G2 actually vary depending on whether you are from a Bantu tribe, a Nilotic tribe, or Kosian tribe, which I thought was really quite interesting. Now, the samples actually vary quite dramatically in terms of sample size, but what's important to recognize is that you could carry 1% in your population or 24% depending on where you're from. And so this really points to the fact that you need to have large populations in order to understand what's going on in terms of APOL1. So just to go to H3 Africa Kidney Disease Research Network, we had a comprehensive phenotyping of 4,000 cases, 4,000 controls. We were also interested in some translational research and capacity building, and I'll highlight that later on, but I'm going to go over the 4,000. We recruited a total of 8,031 participants across 3 countries. We have 23 participating investigators at 9 sites in 4 countries. We have an extensive biorepository with greater than 50,000 samples. And our main thing is capacity building. And I'll say that over and over and over again, that our goal is really to make sure the work can happen here within Africa. We've hosted 11 sessions for training and investigator meetings for more than 420 attendees, trainees, coordinators, site PIs, and collaborators. So I'm going to present the first set of data from our first 3,500. Our genotyping will be complete in January, and our hope is to have our first main paper out next year. But based on our preliminary data, so you guys are the first ones who get to see it, when we take a look at the cases and controls, we have about 1,200 who've got kidney disease 1,800 who don't have kidney disease. We have approximately 35% from Ghana, 43% from Ghana, 57% from Nigeria. The mean age is actually in the 40s, which I think in the previous session talked about kidney disease happens in young within Africa. And we have predominantly females within the cohort itself. The blood pressures are slightly higher in the cases and controls, not surprising. Hypertension occurs in 50% of those with chronic kidney disease. Diabetes is not as common as it is in the United States. Hypercholesterolemia is not that common. And HIV/AIDS only accounts for about 15% of our population. But what you can also see that's interesting is sickle cell disease. Of the patients that we selected was only about 2%, and sickle cell trait was only about 21%. So relatively low, and it may have accounted just for the population that we are recruited from. When we take a look at the allele frequency among the 3,000, the frequencies actually are very similar to the distribution that we see in the US. But what's important to note is that 26% carried 2 risk alleles overall versus 74% that don't carry any risk. And if you're in the cases, you carry 30%. And if you're in the non-cases, the general population, it's 23%. So not as high among 3,500 participants. So it's important to note that this is probably the number that we'll sit at, but it's pretty interesting that it's much lower than expected, and I think we're all a little surprised with that. The other thing is that when you take a look at your definitions of chronic kidney disease, we actually have multiple definitions. The definitions include less than 60 mils per minute, GFR, an albumin-creatinine ratio greater than 30 milligrams per gram, or those who have diagnosed glomerular diseases. When we take a look at adding all of those and say that's our definition of CKD, if you are a high-risk carrier, you're 1.45 times more likely to have a higher odds of kidney disease compared to the general population. And we see that, in fact, it doesn't vary that much between G1 versus G2. If you take a look at those who only are defined as having chronic kidney disease, less than 60, you actually see that your odds ratio is slightly higher at 1.48. But what's interesting is that your G2 association is a little bit higher at 2.62. So there might be some differences that occur between G1 and G2. And if you take proteinuria alone, it's not as highly associated as it is for those who've got advanced chronic kidney disease. So we think among a relatively young adult population that, in fact, your risk is probably close to 1.5 association with chronic kidney disease if you're a high-risk carrier compared to a low-risk carrier. Now, what's important is people want to talk about subgroups and what's the difference, for example, between Ghana versus Nigeria. So for the people in the room, there are lots of differences between Ghana and Nigeria, but there's none of them different by APOL1 status. So what was interesting was the association wasn't any different. It looked like there might be a higher risk among the Nigerians, but it really wasn't significant. There was no gender differences. Among the non-diabetics versus the diabetics, it also seemed that there wasn't much of a difference. And if you had sickle cell trait or sickle cell disease versus no sickle cell, it actually wasn't any different. So now in a much larger population, we've been able to show that there's no interaction with diabetes, no interaction with sickle cell disease, and no within-country differences. So if we were to actually look at the map, odds of chronic kidney disease now would be about 1.6 in the West African population. Much lower than people have reported before. Again, I think we just had power because we're taking a look at 3,500. And just to note that we will have 8,000 within the next few months, and I suspect that we'll have the same data. Thank you very much. Another project that wasn't done by us, but was also done within Africa, which was recently published in KI Reports in 2019. This was actually done in the Democratic Republic of Congo. And this took a look at 412 children from the general population, 401 children who were HIV infected, and said, let's actually measure APOL1 and take a look at what albuminuria is. And what's interesting is if you look at the far right, APOL1 risk was actually associated with albuminuria, and actually in the general population, you're 2 times more likely to have albuminuria in these children. It was sort of a borderline association. And then if you take a look among HIV, it was significant where you're 20 times more likely to have evidence of albuminuria. So if you take these studies together, I think it shows you that depending on your size of your population and what your study question is, your association could actually vary depending on what you're looking at. There's clearly something different about HIV, which I'm not going to discuss today. Sarla is going to talk about that next, and she'll be able to go over that with you in much more detail. But there is clearly something different with HIV disease versus having albuminuria or CKD. So now we really want to understand what are the environments that are associated with development of CKD. So what's important is that when you actually get APOL1, what you do is you actually prevent the virulence factor of the trypanosomal being absorbed into the trypanosome, so you don't get lysosomal swelling of the parasite, and the parasite doesn't die. So that's how it actually, APOL1 itself, though, allows that to be absorbed, doesn't allow it to work properly, so that it actually causes the lysosomal swelling. And so that's how actually APOL1 is protective against trypanosomal infection, rhodesiense. But it isn't associated with gambiense, which occurs in West Africa. So going back to the slide again, it really doesn't quite make sense with trypanosomal infections. And so if this was the reason for selection, it's really unclear why there was a selection of APOL1. Given this difference of APOL1, G1, G2 frequency difference and trypanosomal infection difference, it doesn't seem to really be related. What's also interesting is a group within the H3 collaboration called Trypanogen have actually studied trypanosomal infection in the DRC, Ivory Coast, and multiple sites across Africa. It's a rural disease to get sleeping sickness, and so they've had to go in and collect these cases. The cases have happened years ago. They've had serum that's stored for a long time. It's important to note that overall the cases are really coming down significantly, and there are not that many cases that are reported now. Gambiense, which occurs in West Africa, the reservoir is actually human. It leads to a chronic disease, and it's about 1/5 of the prior rates that there used to be there. Rhodesiense, which occurs in East Africa, is an acute illness. The reservoir is actually animals, but there are less than 100 new cases per year. So what they actually found, and this is quite complicated, as I'm not an infectious disease doctor, and I'm not a sleeping sickness expert, but you can see that in the top row for G1 and G2, that in fact you have this risk for chronic kidney disease. But for some reason, if you carry G1, you are protected against gambiense infection, and you're protected against gambiense infection, but if you carry G2, it's actually orange, and you're at increased risk for becoming a chronic carrier state. So before, when it was just so simple in people's minds that this is actually going to protect us because of sleeping sickness, and that's why it's being carried, and that's why it's related to sickle cell disease, it's the same kind of story. The story is actually evolving much more in that there are differences by G1 and G2, and it actually seems that the G2 distribution, which is important to note, actually is related to the type of infection that you have. And it may be more, it's only protective if you carry G2, not G1. But if you carry G2 and you've been exposed to gambiense, it actually puts you in a chronic carrier state. So there are differences that occur depending on what kind of trypanosomal infection you have. So there is potential that maybe it's involved with some innate immunity. The other thing that was interesting was in East Africa, this was recently published. If you take a look at the different samples they have, doesn't matter whether you're looking at rhodesiense or gambiense, the APOL1 risk alleles were not associated in any way with any protection whatsoever. So there was no benefit in the current day population. Could there have been a benefit 60, 70, 100,000 years ago? Potentially for some selection. But currently, it doesn't seem that there's any true benefit with APOL1. It is in contrast to a study about the same sample size that found that it was protective, but clearly sample is everything because I think we're finding inconsistencies in all the small studies, and so it really behooves us to work together to try and increase the sample size. So what do we know so far? Well, we know that there's clearly some innate immunity that occurs with APOL1 and trypanosomal infection, and how that innate immunity actually protects us, we still have to sort out. But it may be related to HDL function overall, that HDL itself as a protein may actually have some protective function that we need to understand better. We also know that within the glomerulus itself, that it's been associated with increased interferon, with autophagy, with mitochondrial activity, and there's a number of factors that are upregulated. So there's lots of work to go on to really understand why APOL1 actually leads to chronic kidney disease. So the last thing I'm going to talk about is, do we have the right disease? You know, we've been focused so much on chronic kidney disease, and the question that I would say within Africa, perhaps it's not the right disease. And so This was actually postulated by Ned Carney, who's actually a junior faculty member at Sinai, that APOL1 risk alleles give you a second hit, you then get a second hit, then you get chronic kidney disease, and then you get hypertension. Or do you get APOL1 risk alleles, then get hypertension, then get a second hit, and then get chronic kidney disease? Or in fact, does APOL1 cause chronic kidney disease and increase high blood pressure, and you have to get exposed to the second hit to get chronic kidney disease? So just to take a look at the first one, we took a quick look in studies that we had. This published in New England Journal now a number of years ago, where we took a look at the ASK population, which I highlighted earlier, and the chronic kidney disease network called CRIC, and took a look at progression of chronic kidney disease, and said, can APOL1 differentiate between the two? And they are able to differentiate with progression over time. But importantly, we tried to see if there was a difference between proteinuria and not. So if you had chronic kidney disease from hypertension, was there a difference? But most importantly, if you were treated with intensive blood pressure control versus standard blood pressure control, and I stratified the analysis by your APOL1 risk, there was absolutely no difference. So actually treating patients with APOL1 with more intensive therapy when your creatinine's already elevated didn't make much of a difference. And it didn't make much of a difference if I gave you an ACE inhibitor or a calcium channel blocker too. But remember, those are people who've got chronic kidney disease who then go on and develop hypertension. Now the question is, what about those who've never had kidney disease before? And in a large sample of a few thousand that were screened at Mount Sinai, what this shows is that actually if you take a look at your left, you're taking a look at the age group of the bottom sort of x-axis from 20-year-olds to 40-year-olds. And you can take a look at systolic blood pressure in the top panel, diastolic blood pressure, estimated GFR. If you're at higher risk, it's actually in the red, APOL1. And if you're in the blue, you're the low-risk alleles. So you can see there are slight differences that occur in your systolic and diastolic blood pressures by APOL1 risk. And then I sort of blew it up there on the right so you could see a little bit closer, that there may be a difference in the young adults, in the very young adults, especially the 20-year-olds perhaps, that there's a difference between blood pressure control. In the analysis that they did, they took a look at the homozygotes, which were in the red, versus the normal controls, and you could see that in fact, sorry, blue the other way is blue is the risk. Those are the ones in higher risk, so you can see if you are younger, you're at actually higher risk for having hypertension than those who are older. So they actually hypothesized that in fact, if you were to stratify young adults, the 20 to 39-year-olds, that if at wild type, that means you've got low risk alleles, there was no change in blood pressure. The heterozygotes actually had a slightly higher blood pressure, 1.5 to 1.6 millimeter increase. But in fact, those who carried the two risk alleles were at the highest risk. So they were at 2.5 to 2.9 millimeter mercury increase, which may not seem like a lot, but if you're a 20-year-old and every year you're going up by 2 millimeters of mercury, which was surprising, and this was followed over decades. And in West Africa, in our H3 Africa population, in fact, what we found was that there wasn't much of a difference if you look at the two panels on the left of systolic blood pressure and diastolic blood pressure. APOL1 homozygotes are solid, and then the risk alleles are basically in the dotted line. So the dotted line is increased risk, and the black line is low risk. And you can actually see that there's no difference if you have kidney disease. But in fact, what's so interesting to me is that there is a difference among the control population. So potentially, in young adults who have no evidence of chronic kidney disease, there might be a difference with APOL1 risk that's important that we need to try and tease out and see if that's consistently an association. So there's really no shortcuts on the long road of evidence to evidence-based medicine. I know that people are all, in North America, everybody wants to genotype everybody and test APOL1. I am highly skeptical yet that we should do it routinely without us knowing what are the potential factors that lead to progression if you are an APOL1 carrier. I also realize that within the continent, we need to actually train more people because this is the continent that's going to be affected the most by actually understanding APOL function. So training, so Charlotte Asafa and Yemi Raji actually did Masters in Human Genetics at University of Michigan. They graduated in 2017. They actually survived the snow in Michigan for a year and did extremely well. Charlotte's actually now funded by a K43 Fogarty grant to do research in preeclampsia and APOL1 in West Africa. So sometimes, even if I stand in the middle of the room, no one acknowledges me. One of the things that's really important for us is actually community engagement also. I think it's really important that we try to explain to patients, ultimately, what is your APOL1 status, and what does that actually mean, right? Patients get what their blood pressure means, and they get what their weight means, and they get what their cholesterol levels mean, but people aren't quite ready yet to understand what their genotype means. And so there's lots of community engagement going on with the H3 Africa collaboration, and Ifeoma is leading this also with Dr. Adu in Nigeria and Ghana, and the hope is that we'll understand a little bit better about what we're doing in terms of reporting back to patients. So I'm just going to end here to say that high-risk carriers occur in 26% of West Africans in Ghana and Nigeria in our 3,500 participants. There are lower rates in East Africa, but it's not a large enough population yet. And we really need larger cohorts from also Central and South Africa in order for us to really understand APOL risk across the continent. I think APOL1 may be associated with blood pressure in young adults without kidney disease, and we need to understand that much more. But there's no differential effect of sickle disease trait and APOL and diabetes, which I think is important. And hopefully we'll have a final paper to present next year to you. So this is what we really need to do. We need to study gene-gene interaction, gene-environment interaction. And all together, our hope is that we develop these large cohorts of chronic kidney disease across the continent, understand interactions, understand HIV disease, trypanosomal infection, understand if routine screen is actually impactful. Should we routinely screen for blood pressure? And what are the second hits? And this is really sort of our work over the next 10 years, I think. Lastly, this is the group that we all work with again, and I can't thank them enough for all their work, and to thank them for letting me present today on their behalf. This was our last meeting in Ibadan, and as you can see, we've grown enormously, and we really think that we have lots more to do. I'm going to end with a phrase that I heard this morning. I don't know if Bashir is here, but Bashir said this: African problems need African solutions. I think that's incredibly important, and I think this is just showing you how important it is to study genetics within the continent, and my hope is that we'll continue to train people so you can solve the problems. Thank you.